Okay, welcome everyone to this year's annual BPhO lecture, sponsored by G Research. It gives me enormous pleasure today to introduce a friend and colleague, Professor Sarah Bohndiek. I've known Sarah since we were students together in Cambridge. She's actively said to me, do not give a large blurb, and in the interest of time, I'll just say she is one of the world experts in medical imaging. She's currently a professor of biomedical physics, and I'll give you the floor, Sarah.
Thanks very much, Anson, for welcoming me here to the lecture today. It's a real pleasure to be able to talk to you all and to be in a room full of such budding physicists. So my talk today is going to focus on how I'm applying physics for early cancer detection. But before I get started on that, I thought it might be of interest to some of you to know how I got to where I am today. So I grew up in southeast London, and I did my GCSEs at Thomas Tallis School in Kidbrooke in London, a comprehensive school which has since been knocked down and rebuilt. So the picture that I show you there is nothing like what it looks like today. I then did my A-levels. I did physics, maths, further maths and history at Sixth Form College in Lewisham before going on to Cambridge to study natural sciences. So for those of you who don't know, natural sciences in Cambridge gives you an opportunity to explore a lot of different sciences in your first year. So in addition to physics, I also took material science and evolution and behavior. Now, that might sound irrelevant to you now, but growing up, when I was doing my GCSEs and A-levels, I wanted to be an astrophysicist. I grew up by the Meridian line in southeast London. I was obsessed with astronomy. I did an astronomy GCSE. I used to go to astronomy camps. And in my first year of undergraduate study at Cambridge, when I was going to material science lectures, we had a whole series of lectures on biomaterials. And I have to admit, I hated material science. I wasn't studying chemistry. I didn't have a clue what was going on. But I really liked the biomaterials lecture because it gave me the first glimpse of what it would be like to do research which could actually impact people in their everyday lives. And that almost completely turned around the direction of my career in those early undergraduate lectures, because I went off to do a PhD following that in radiation physics in University College in London in their medical physics and bioengineering department. And during my PhD, I spent most of my years in a dark room characterizing what were then called CMOS active pixel sensors. And these are basically the cameras you find in all of your smartphones every day. And we already heard from our colleague from MPL earlier about the importance of making new measurement devices to understand color. In this particular aspect, I was really doing the most boring thing possible. I was taking pictures of completely uniform fields of light, so completely blank screens, to characterize how well these different cameras were performing. And then I applied them to look at X-ray diffraction profiles that were taken from different types of breast biopsy samples. So that was the medical angle in my project, was devising a new way to make measurements of the presence of breast cancer using X-rays. And as I moved through this, I found that quite a lot of the features that we were looking at were really the presence of different biomolecules, which are based on these X-ray diffraction profiles that we were measuring. But as someone who'd really focused on physics and loved physics for most of my childhood and my adult life to that point, I really had no idea about those biomolecules. What was the underlying biology in the first place? And so I decided when I finished my PhD that I really wanted to just jump across to the other side. I wanted to understand what biology was all about. So I went to work for a few years back at the University of Cambridge in the biochemistry department. So this was a completely new experience for me. My role as a physicist was to fix the magnetic resonance imaging system whenever it broke. I would be there tinkering in the background, trying to work out why the vacuum had lost or why the helium was boiling off and all sorts of things. But in return, the group taught me how to do biology. So I was there working for the first time with laboratory animals, inducing cancer models in mice, running enzymatic assays on different cell cultures. And the work that I did there showed how we could use vitamin C, a relatively simple, ubiquitous biomolecule, to measure metabolism in cancer. And these are just some examples of the ways that we did that by isotopically labeling vitamin C with carbon-13, replacing the intrinsic carbon-12. In the kind of fullness of time, I realized that it was really interesting to get an international experience, having spent all of my life so far in the UK. And one of the places I really wanted to move to, having fallen in love with California during holiday visits there, was Stanford. And I spent a couple of years there with Professor Sam Gambier, who's one of the pioneers in early cancer detection, and was also trained in mathematics, by the way, before doing a medical degree, and ended up really having a foot in two camps. And during that time, I was still using optics, still working with cameras, but this time imaging different ovarian cancer models and looking at how we could use that for early detection. So what I really want to say to the kind of the culmination of this is that 2013 is when I finished my postdoctoral work and then went on to set up my own laboratory in Cambridge. So 11 years from those of you who are finishing A-levels, 11 years from finishing my A-levels, I was in Cambridge setting up my own lab, doing research independently, and raising my own funding to find new knowledge and generate new knowledge. So it's really something that's not that far off in the long distant future for you, if that's a direction that you're thinking to go down from a careers perspective. What do we do in my lab? We develop and apply different tools from multi-spectral imaging, and we combine those with computational analyses to study the early phases of tumour evolution. Now, first, let's think what multispectral imaging is. This is a multispectral imaging montage of the sun, and this is taken by NASA. This multispectral view shows you what happens if you look at the sun in different narrow spectral bands. So we select different colours, here in the image going from 717 nanometers in the red, all the way through to 9.4 nanometers, far down in the deep UV. And if we look at the sun in those different colours, you can clearly appreciate that you see different contrasts between the different areas of the sun. And those of you who've got the prizes related to astrophysics will be much better at telling me what that all means. Now, in my lab, we use multispectral imaging for a different purpose. We capture different colours of light in order to detect the underlying colour of cancer. And we do that in different ways. We use tools such as photoacoustics, where you combine light with sound to get deep tissue penetration with optics. We also use endoscopy, which is primarily what I'll talk to you about today. We advance different innovations from optics and photonics, which may be used for remote sensing or telecommunications, and bring them into the medical realm. And a significant part of what we do is really modelling those light-tissue interactions and understanding the processes that the light has undergone as it's passed into the tissue, where it's been absorbed, where it's been scattered, and then inferring from that what's actually happening in the biology. Increasingly, we also use a lot of tools from machine learning in our lab, and that's something that allows us to interpret some of our data based on physics-driven simulations. And we apply these different tools in preclinical studies in cancer models, which I've already alluded to, and also taking them through to first-in-human clinical trials. So what I'll focus on today is just one subset of the research where we're looking at endoscopy, taking new innovations in optics, and applying those in clinical trials. So where I'll start is by telling you why I think optical imaging is powerful in the measurement of hemoglobin in our red blood cells and why that's relevant in early cancer. I'll then introduce a couple of different ways that we've brought about that allow us to make these sorts of colour-based measurements in an endoscopic setting. So actually putting new colour cameras inside patients to extract this data, and then show you some of the results that we've got from some first-in-human testing and why we think this is exciting going forward. I'll start then from a little bit of cancer background. Solid tumours, as they form in people, require a vascular network to obtain oxygen, nutrients, and then remove metabolic waste. So when we're early on in the development of cancer, which hopefully we can then see the laser pointer there, we have an avascular mass, so that the originating cancer cells have mutated, they've gone on to proliferate uncontrollably, and they're growing and they're growing and growing, and eventually the cells in the middle of that mass are sufficiently far from a surrounding blood vessel network that they reach a state which is called hypoxic. So they don't have sufficient access to oxygen or nutrients in order to undertake their normal metabolism and to support their really fast growth. When that happens, then start to secrete what are called angiogenic factors. So they secrete biomolecules that are passed through the interstitial space to nearby blood vessels and stimulate those blood vessels to leave their usual healthy quiescent state and start to grow themselves towards the tumour mass. And this, over the course of time, then leads to a tumour vascularization. Now, the interesting thing about that process is that the tumour vasculature is then completely different from our normal healthy vasculature. In a normal human being, we have a healthy arterial supply and venous drainage of blood, and we can perfectly meet the metabolic needs of a tissue at any given time. However, in a tumour, because of this kind of chaotic distribution of blood vessels that occurs as it starts to stimulate growth factors in different directions, we end up in a case where the vessels that grow in are inefficiently oriented, they're poorly perfused, and we have different limitations on the delivery of oxygen, including perfusion limitations where the blood doesn't flow in sufficiently fast, as well as diffusion limitations where the oxygen unbinds from hemoglobin and just can't diffuse far enough out into the tissue. And what we know from a biological perspective is that oxygen starvation leads to hypoxic stress, a word I already used, and that hypoxic stress, so the stresses that occur in the cells due to the absence of oxygen, leads to really poor outcomes in patients. And this is just an example in breast cancer patients where survival is much poorer if you have a high level of hypoxia in your tumour. Now, the case study that I'm going to use in this particular presentation is Barrett's esophagus. And this is a precursor condition that can progress through different early stages of cancer to a deadly cancer, which is esophageal cancer. Now, these patients are at risk of developing esophageal cancer, and at the early stages, this risk is relatively low. But if their tissue undergoes mutations and early changes towards these intermediate cases, which are called dysplasia, their risk of developing cancer increases dramatically. And esophageal cancer is particularly deadly with only a 20% 5-year survival. So what happens in these early stages is that we get changes in the vascular architecture and we get changes in the blood supply even before the presence of cancer. So if we could visualize that non-invasively, we would be able to identify these dysplastic lesions and curatively resect them. So the endoscopist could go in just with their regular biopsy forceps for cutting the tissue and cut out these suspicious regions and prevent the person from going on to get cancer. The difficulty is that at the moment, these early changes are incredibly difficult to find. So I already mentioned the word endoscopy. What do I mean by an endoscope? So this is just a schematic example to show what happens during endoscopy. This is a colonoscopy, but equally these can be used in the esophagus. So a long, thin tube containing a camera at the tip is passed into the patient. And this camera is incredibly small, but incredibly high resolution, of a similar type to that you find in your smartphones. And there are some light sources there to illuminate, because obviously when we're inside the body, there is no other source of light. And these are the forceps that I mentioned that come down to cut out the tissue. Now let's think about this camera in a bit more detail. These cameras are made out of individual small pixels. If you've bought a smartphone, then you'll know that your camera has X number of megapixels, and more megapixels equals better. Each of these megapixels is an individual electronic sensor. That electronic sensor is made of silicon, and when light falls upon silicon, you generate electron hole pairs, which are then captured by putting that pixel under an electrical bias, and we can measure the electrons that are accumulated in that pixel, and they give us a brightness measurement. So each individual pixel on that camera has an associated number of electrons, and more electrons is brighter, means there's more photons have fallen, and fewer electrons is dimmer, means fewer photons have fallen. And what we want to do is then measure these in a way that allows us to look at the colour of the tissue. And in everyday life, when we're looking at smartphones, then that colour comes from the application of individual tiny little optical filters on every single pixel. The colour of those filters is determined by the colours that our eyes can perceive. So the red, green and blue cones in our eyes are the ones that are able to respond to those colours. So these are the colours that we see and are able to perceive, and that's, although we only perceive these 3 colours, we can then compose the rest of a colour scene from these. Now the question then comes, how does that become a colour photo? So if each of these individual pixels is measuring the brightness of red light falling on it, green light falling on it, or blue light falling on it, where do we get a colour image? Well, if we take a nice example here, just a simple image of a scene. This is a raw photo. It's just a grid of coloured squares. First, we take all of the pixels that are red and we make a red image, all of the pixels that are green and we make a green image, and all of the pixels that are blue and we make a blue image. But of course, there's then missing pieces, so we then have to interpolate computationally what's missing, and then we merge these together in order to make a typical colour scene. So that's how a typical colour camera would work, and that's what's currently used in endoscopy. Now, what I'm going to explain next is how blood absorbs light, and then get you to have a look at some endoscopy pictures. So if you're squeamish, I'll tell you when to look away. So hemoglobin within red blood cells is a strong absorber of light, and this is how the absorption of light varies as a function of wavelength. And what's really interesting here is you can see that not only is blood strongly absorbing, it's also differently absorbing depending on whether oxygen is bound or not. So that gives us a way to measure not only the presence of blood and vessels in a tumour, but also how well oxygenated it is, or conversely, how hypoxic it is. The other thing you'll notice from this plot is that it's extremely absorbing in the blue and green and much less absorbing in the red. So that's why when we look at pictures of our skin and inside the body, we mostly are seeing changes where we are getting really strong absorption of red and green light and less absorption of red light. So let's have a look at some endoscopy pictures. So bearing in mind the fact that if we look through an endoscope, our esophagus is mostly going to be absorbing blue and green, where it's perfused with blood, and much less where in the red. So here are some pictures of a classic Barrett's esophagus patient. And I'd like to see if anyone can guess where the lesions are. If you'd like, just give you 30 seconds to have a look. Put your hand up if you feel really confident that you spotted the cancer. So about 0.5% of the audience are really confident that they know what they're doing. So you can tell this is a really hard problem, okay? So in the first image, we've got a lesion over here, in the second image over on the left there, and in the final image over on the right. So these images are looking pink because the red light's being strongly reflected and the blue and green light's being strongly absorbed. And this leads to relatively poor contrast for cancer due to this relatively simple perception phenomena. So if we look at these 4 groups of colours here, we've got some groups of reds, groups of yellows, groups of greens, and groups of blues. Now, if I get you to look at all of these, you can probably very easily tell the difference between, let's say, the groups of yellows. But you might find it quite hard to tell the difference between the groups of greens and the groups of reds. If we actually look at the spectrum, these colour panels have exactly the same difference in terms of their wavelength. So the red separation is actually the same wavelength separation as it is for the blues. But we're much better able to perceive differences between blues and greens than we are to be able to perceive differences in reds. And that's why it makes it really hard to spot cancer. So if we could go beyond our colour vision and overcome some of the limitations, because we are only human after all, we might be able to improve the visualisation of these early cancers. So one example is that already is present in the clinic, is looking at depth resolution. That's something I won't talk about today until right at the end. An example in terms of looking at colour is something called narrowband imaging. So instead of shining light on the tissue and just sensing everything that comes back in this wide range, in narrowband imaging we focus on 2 narrow colour channels which are associated with strong optical absorption of haemoglobin. And these pictures, this is a standard white light picture on the left, and this is a narrowband image on the right. So immediately it looks like there's a whole lot more contrast there. And when we zoom in on the lesion region, there's actually a lot of interesting features here relating to the vascular architecture, which are highlighted by the fact that we're using a wavelength that's really strongly absorbed by blood. What we then thought in our lab was, well, instead of maybe just using 1 or 2 colours, why don't we go wild and measure an entire colour spectrum from the tissue? And that's where this concept of hyperspectral endoscopy or hyperspectral imaging comes in, which is instead of taking just 3 colour images when we're taking pictures of a scene, let's take an entire spectrum at every single pixel in the scene. And that would allow us to better classify the disease because we'd be able to measure these spectral changes that are being provided by the absorption of light by haemoglobin. Now, the challenge there comes with miniaturizing something to a point where you could actually integrate it into an endoscope because most spectral imaging devices are large benchtop machines that are very expensive. If we were thinking about how to do this, there are several different approaches. Now, I already mentioned that there are these cameras on the tip of endoscopes, and these are called chip-on-tip endoscopes, and they're pretty ubiquitous now in hospitals. But there are also endoscopic configurations called baby scopes, and these are fibre optic bundles that you can place down inside the accessory channel, which is the hollow tube in an endoscope. Now, we typically don't use these very much from a clinical perspective, but those accessory channels are present in all scopes so that the endoscopist can put their biopsy forceps down. Now, instead of putting the biopsy forceps in, we can put our optical fibre bundles in, and we can make measurements of the spectral characteristics of the tissue directly in situ in patients. And that's the approach that we've taken in terms of a research perspective, because we can do a very good job at acquiring very high quality spectra and understanding how those blood profiles change at those early phases of disease. So that's something that wasn't known. It was kind of well established what the vascular changes were in the advanced disease. But when we started this project a few years ago, it was not well known what was happening in those early phases of dysplasia where one can actually intervene and cure. So we started with this babyscope approach, but in parallel, we also were developing new ways to create colour filters for cameras that would allow us to ultimately replace the standard of care. Now, the reason for doing this is one that might seem very far from your physics realm, which is that the regulatory burden of actually creating a new medical device and getting it into a hospital is a time frame of something like 10 to 20 years. So if I have a great idea and my device works really well, it's probably still going to take me 10 years to have a commercial product in a hospital, which means that the testing phase is much more efficiently done with existing products that are already in hospitals. So strategically, from a research perspective, if we want to gather this data from early stages of disease, it's better if we can use products that are already clinically approved. So that's where the babyscope approach came in. So there are already clinically approved fibre optic bundles that one can use for testing in patients. They are used in a range of different disease categories. And we took these existing approved devices and we introduced these imaging fibre bundles into them. They're designed so that there's a glass tip for sterility, so you can have a disposable catheter that you put your fibre optic bundle into. And this entire thing goes inside an existing endoscope. So we can make it whatever measurements we want, but we're not changing the standard of care. The bit that touches the patient is still clinically approved. And this is all done at our local hospital in Cambridge. Now we want to, from this fibre optic bundle, somehow acquire both spatial and spectral information. So this is a little bit of a challenge. And here's a schematic of how we did it. So this is our catheter coming back here into an imaging channel. So we have an objective lens, which allows us to image the distal facet of the fibre, the proximal facet of the fibre, so the one that's outside of the patient. And this is the bit that would go inside the patient. And so this is the bit where you saw before. You have an illumination fibre and an accessory channel. So we take a picture of this optical fibre, which is essentially a bundle of fibres clad together. And we relay that through an optical device called a beam splitter, where a small percentage of the light, about 10% of the light, is sent to a standard camera, so we can see what's happening as we're moving our fibre optic bundle around in the scene. And the rest of the light is sent into a spectrometer. And this is a device that can then disperse the light. And this is a particularly useful spectrometer because it has a 2D camera on it. So I can take data along one line of an image, and I can disperse that line so that one dimension on my camera encodes the spectral information and the other dimension on my camera encodes spatial information. That means that in order to build up a full image, I have to scan my endoscope around. But it turns out that's what endoscopists do anyway when they're trying to look for cancer. They kind of look around the esophagus. So that's okay. We then have a white light LED to illuminate the tissue, so we're using the same kind of broadband light source that we normally use. Now, the reason that this works so well is that we have our regular high-resolution image, which allows us to reconstruct the scanned information from the spectral camera. Now, normally, when you're waving an endoscope around in a patient, you have all sorts of geometric distortions that come out. You get issues due to rotations, magnifications, things getting closer to and further from the tissue. The patient breathes and their heart beats and all sorts of things get in the way. But because we have this very fast high-resolution camera that's taking all of this data, we can then recompose this very high-resolution image based on that data using just simple computer vision tools. We can then apply the mathematical geometric transformation matrices from these computer vision tools to the spectroscopy data set that we acquire. And that allows us to reconstruct a high-fidelity spectral data set. So here you can see on the left the white light camera data, and here you can see just a small subset of the spectral data. So in this implementation, we acquire data at a few nanometer intervals, all the way from 400 nanometers to 700 nanometers. And we do this in real time while the endoscope's moving around in the patient. Now, before we went into a patient, we had to do some very basic testing. So first, we had a look at profiles from biopsy samples that were taken from patients, and we showed that the profile of the normal healthy tissue is distinctly different from that of the patients who had this precursor Barrett's condition, and also distinctly different from the cancerous condition. And we also showed, warning, another squeamish image coming up, that if you take an esophagus out of a pig, so this is a pig stomach with the esophagus attached, and you insert your endoscope into this and wave it around, you can actually capture image data in real time. So now my endoscope's looking down the esophagus of the pig towards the stomach, and the dark region you see in the middle is the junction to the stomach, and all of the data you see around is the tissue. Now, I'm going to switch gears for a bit and tell you a bit about the research and development on the chip and tip side before I introduce some of the human trial data. So let's say that's all well and good. We can go and collect our spectral data, but we still need a camera that would allow us to capture this different color information in a clinical setting. But what could we do? Well, we could take our existing color filter array that's red, green, and blue, and we can just throw that out and say, let's pick some other colors. Let's pick 9 or 16 or some other arbitrary number of colours that are targeted towards the relevant colour changes that the presence of haemoglobin induces and how those are being done at the early stage of cancer development. Now, the problem with doing that is simply that actually creating optical filters at the pixel level in a customised way is extremely difficult. In a conventional colour filter array, you manufacture using either absorptive dyes, one for each wavelength, or using multi-layer cavities, which are combined from combinations of different alternating dielectric materials for each wavelength. Whichever one you use, you have to have a different one for each different color that you want to measure. So that means when you're going through a process, we typically use a process called lithography to do this. So we either expose our, create it using photolithography, which uses light, or electron beam lithography, which uses electrons. In the standard smartphone setting, this would be photolithography done on a huge scale with billions and billions of devices a year. But it's very cumbersome if you want to do more than 3 turns. So here I want to make 3 different colors, so I have to have 3 different masks to block off the exposure dose in different regions and allow me to deposit green filters on a particular pattern, red filters on a different pattern, and blue filters on another pattern. So now if I want to do this 16 times, I have to do 16 steps. I have to take my thing, put one filter down, go again, put another filter down, go again, get another filter down. And so it becomes incredibly complex, and no one wants to make these things. It's just not economically viable to do it for different approaches. So what we had to do then was go back to the drawing board and create a whole new lithography process that would allow us to customize these optical filters. Now, this process is based on a relatively simple principle, which is that of a Fabry-Perot cavity. This cavity is one where you have 2 mirrors, and the wavelength of light that passes through this cavity is simply described by the distance between the mirrors and the angle of incidence. So this is very nice. It's a controllable cavity where instead of the color of light being defined by an absorptive dye, now it's defined by the thickness of the cavity. So if I have very small, thin cavities, I will couple light towards the blue region, and if I have large, thicker cavities, I will couple light towards the red region. Now, the clever thing comes with how you actually make these cavities. It's not new to put this type of cavity on a camera. What's new is to do it in one single step. And the way that we do that is by varying the amount of exposure to either the photons or the electrons that you would get in this lithographic process based on what's called the exposure dose. So high exposure dose means that we get a lot of electrons and photons present, and low exposure dose means we get relatively few. So the data I'm going to show you are from a photolithography process, so this is either lots of photons or very few photons. And the material that defines the cavity height is a material that is photosensitive. So if it gets lots of photons, then it will be resilient to chemical stripping. So it will hang out there no matter how much chemical I put on top of it. Whereas if it gets very few photons, it doesn't have the same resistance, and it just gets washed away if I put lots of chemicals on it. So by patterning with different amounts of photons at different positions on my camera, I can then define cavities of different heights when I apply chemicals to strip off the plastic that's left behind, the polymer that's left behind. And what that looks like in practice is that, so here's a dose matrix of how much dose has been given, and then here's an atomic force microscopy image of how tall of each of those cavities were that was left behind as a result. And the nice thing about this process is that it's very flexible. So now I can do arbitrary colors. I can do the standard kind of red, green, and blue color imaging, but I can also make arbitrary multispectral arrays. And one thing that we've done most recently is to start to nest small filters within a standard blue filter, green filter or red filter to allow us to do spectral sampling but still recover standard colour images. So let's then think how we might take this idea of capturing colour information and actually test it in people. Well, you know, it's very easy to think about the pipeline for doing this, but there's a lot of work that has to go on before you can put it in a person, obviously. You want to make sure it's safe, you want to make sure it's effective, you want to do tests in those kind of pig models, in what we call phantoms. These are not ghosts, these are just test objects that look a bit like people, mostly composed of jelly containing milk and coffee, and that weirdly turns out to look pretty much like a human if you're an imaging system. So we've done all of those sorts of tests and things look pretty good. So we set up a research study with one of my collaborators, Dr. BPhO, who's a gastroenterologist at Addenbrooke, to assess the feasibility of actually doing this in endoscopy and to evaluate the sorts of patterns we might see in abnormal tissue versus healthy tissue. So how do we get this device into patients? So we go back now to our baby scope. We have our baby scope. This is the standard endoscope. We then have our little trolley that contains our endoscope that goes down into that accessory channel and into the patient. So it looks a bit like this. It protrudes out of the accessory channel into the patient. We can then give a data display to the clinicians showing the spectral information, but as I'll explain, it's not really what the clinician wants to see. We send light down into the endoscope, we collect light coming back, this is our setup with the camera, and there's spectrometer that disperses the light into different colours. And then we have the endoscopist move their scope around so we can pick up the spectral data. We then set it up so they would target particular areas for imaging. So the endoscopist would look under their standard white light image, and they'd say, this particular region looks a bit suspicious. Then we would introduce our endoscope so that we could image in that suspicious region. And then they would introduce their biopsy forceps and actually cut it out. And so that means that we would have a reference histopathology report where someone has actually cut up the tissue and stained it and looked at it under a microscope at very high resolution to check exactly what cells are present. So are these normal, healthy esophageal cells? Are these Barrett's cells? Are these dysplastic cells? Or are they cancer cells? And then we would be able to register our spectra with the gold standard pathology report. So the first thing to note is that our gold standard is imperfect, so the endoscopist and the histopathologist can get it wrong. So this is one of the huge problems for physicists making new medical imaging devices, is that often our devices are better than the humans that are taken as the gold standard. And it's really impossible to prove that because you can't prove you're better than a human if the human is imperfect in the first place. So when we're developing our work, we try to only look at examples where the endoscopist and the pathologist agree, because hopefully 2 heads is better than 1. And in that case, we first look at the variance in our data. So it's really important, as you'll know from doing experimental physics, that you don't just do a measurement once and then hope that's the best it's ever going to be. You never have an N of 1. You always try and repeat your experiment and try to understand where the variance is coming from. So we repeated our experiment in many different patients. The data you're seeing here is from 15 patients from which we had gathered over 700 spectra. And the healthy tissue, which is called squamous here, and the Barrett's tissue is highlighted in blue here, and then any unusual tissue is, that's suspicious, either cancer or dysplastic, is shown in pink. So what you can already see is that there's much more variance in the diseased tissue states than there is in the healthy tissue. So between patients, our healthy tissue, so between all of us, our healthy tissue looks quite similar. But if we develop disease, our diseased tissue is quite unique to us. The other thing to note is that we can then go into this in a lot more detail, so look at the statistics of how things vary, and we can see that we get even more variance in the cancerous tissues than we do in the abnormal Barrett's tissues. The next thing that we do is actually look at the individual spectra and try to fit biophysical models to these to understand what biomolecules are governing them. Fortunately, our hypothesis was right, that the spectra that we measured are governed by the absorption of light by hemoglobin. You know, a few could have been something completely different, but we got away with it. So we have changes in oxygenation and we have changes in vessel size, and these are modelled spectra computed using a model of tissue optics, which shows us what we would expect the attenuation of light to be in cases where we have different oxygenations ranging from 0 to 100, and when we have different vessel radii ranging from very fine to very large. And then we can look at those fitting results, and we can see actually in this particular case, the oxygen saturation didn't change very much, which was contrary to our hypothesis, but the vessel size and the vessel blood content both changed a lot between early stage disease and later stage disease. And this is, we think, due to the biological fact that vessels penetrate upwards as disease is progressing, so they get closer to the surface. And our endoscope is measuring from the surface, so we're actually seeing a higher blood content because the volume of tissue that we measure has more blood vessels in it. And we want to test that hypothesis now by actually looking in detail under the microscope at sections of this tissue, and this is an example of one here, that are stained with a stain called CD31, which allows us to look at the presence of blood vessels. And all the blue that you see here is just the presence of nuclei in cells, and it's the brown that we actually want to look for. If you look with your eyes, you'll probably see that similar to my earlier example, it's really hard to see this brown over this blue, but fortunately computers are much better at doing it, so we can develop, again, machine learning algorithms that allow us to classify both the position and the size and various other characteristics of the tissue. The other thing that we did in this example is we looked at how the spectra could be used to classify disease states. And we showed that in classification, we could get relatively good specificity. So we were very good at saying a confident no that there was no disease present, but a bit less good in terms of sensitivity, only 75%. So we have to think about how these spectral information can be used. And one key factor that we had to consider here was actually how a clinician was going to interpret it. So we could use our spectra that we captured in a computational sense to analyze the disease, but we could also use it in a similar way as regular color cameras are used, simply to display something that the human could interpret. One way to do that is actually to use our classification, but instead of classifying to disease type, classify to the nearest visual color that that spectrum corresponds to. And the way that we've done this study is that we've actually looked at the nearest spectral color that you can see in something called a Macbeth color chart. So a Macbeth color chart is a chart of different colors that represent the most frequently seen colors in our daily lives. So they're the colors that are used to test video cameras, if people are out filming for the BBC, for example, and they're used to test a whole host of different camera technologies. So we use them to gather data, to have some reference spectra for different colors, and to see how close our esophageal ones were to these reference spectra. And the sorts of ways that we can then do, sorts of things we can then do are to map the different color classifications over onto our images, so that a clinician can look at their image and instead of seeing, oh, I've got pink and a slightly different shade of pink and a slightly different shade of pink, we're mapping onto different color channels. So now they actually see blues and greens and pink hues for corresponding to different disease states. Now, having gathered all of this spectral data and looked at how it relates to the disease classification, the other thing that we can do is try to identify which colors are most relevant for discriminating between different tissue types. So the way that we did this was to develop an optimization algorithm that would look at what happens if we take 3 color filters, but instead of taking red, green, and blue, we take ones that are best targeted to be able to reveal the contrast for early cancer. So this optimization process, we use some optical simulations where we patch together a pretend case study. So in our case study, we started with white light endoscopy. White light endoscopy uses the red, green, and blue filters. And we patched together a picture, a synthetic picture, which included the healthy tissue adjacent to regular non-dysplastic Barrett's esophagus, in which was embedded some neoplasia. So this is akin to the clinical scenario where the endoscope will come down through the normal squamous epithelium, it will hit a region of Barrett's, and within that region we'll be looking for cancer. And if you do this simulation for the standard endoscopy, you see a color contrast of about 3, which explains why it's so hard to see cancer in endoscopy as we have it today. If we do then narrowband imaging, where we focus down on restricted color channels, this improves slightly. It gets to about 6. Now, if we optimize for the best color bands that are present, we can actually get this contrast to be up to 26-fold between the regular cancer and the background. So if we were to customize our targeted spectral filters, we would be able to much better identify early cancer. So using this colour information, I hope has convinced you, shows some promise, but there's a whole lot of work that you need to do in medicine before you can get these early stage physics and engineering developments to be applied in practice. For example, all the data we acquired so far was just at our local hospital in Cambridge. We would need to do multi-centre validation of these spectral types across different disease populations, so let's say different parts of the country, different groups that are presenting with the disease across different hospitals. We would then need to do what we call ex vivo analysis, so analysis outside of the body, to confirm the properties that we see in vivo. And we would then also need to do this high-resolution microscopy to validate the models that we're using to interpret the data. We would then need to test how this would look in a chip-on-tip camera, and that's a clinical trial that we're currently starting up in Cambridge, and we hope to get going over the summer. So we'll now be doing high-resolution imaging down the endoscope in patients with esophageal cancer, but we'll be doing it at 16 colour channels. We also could test in other disease settings, so we could test in polyps, for example. So I think that's a bit of a summary of where we've got to in terms of our testing, but what I thought I'd end with is maybe just a summary of the presentation and then maybe some lessons that I've learned along the way. So I hope I convinced you that visualizing hemoglobin is important in the detection of early cancer, and particularly relevant in that early dysplastic setting prior to the development of cancer. Those vascular changes are already happening and can be used to make a curative intervention. If we want to measure them, we need to change the colour channels with which we view down endoscope, and we can do that in different ways. From a research perspective, we want to capture all of the colour channels so that we have the most information available to develop future devices. And from a physics and engineering perspective, in order to create these, we need to develop new approaches to optical filtering and measurement, as well as new computational processes to process the data. And with the lessons of the time it takes to get these things to clinic, I think the message from Robin Hughes earlier was really important about perseverance. You really have to have a dogged determination to not only conceive one of these ideas, but then develop the technologies that are needed to measure them, think about the underlying physical interactions and what's causing these from a biological perspective, and then try to get to a point where you can actually measure it in a human. So the sort of results that I'm showing you today are the culmination of only 6 or 7 years of work, but it will probably take another 6 or 7 years of work to get this realized in a clinical setting. I think the key for me in terms of developing these technologies is that I'm really excited by the potential of using physics in a medical environment, and I think that's the most important career lesson that I think I've ever learned, is to just be excited by what you're doing. Be excited by what you don't know, don't be afraid of it, and always be prepared to try, even if that means you have to go and work in a biochemistry department when you're a physicist. It doesn't matter if it doesn't work out. The world keeps on turning, you'll find a new opportunity. And as a physicist who works very closely with biologists and clinicians and engineers and chemists and mathematicians and computer scientists, I can tell you that I am never an expert in anything in my room, and I'm always going to have to ask questions, and there are no stupid questions. You should always ask for help and always take the help that's out there, because you'll never be able to do it all yourself. So from that perspective, make good use of your teachers, your mentors, your role models, and always be prepared to reach out to people to help you to get to where you want to go in your career. For me, most importantly, appreciate those people who stand beside you. So that's for me, my parents, my husband, and now my children, who are always there when I want to reach out and have someone to support me, not just necessarily the teachers and mentors from a scientific perspective, but very much the family that are there for you from a personal perspective. So be brave and seize opportunities and follow your path, even if your path wiggles all over the place. It's long and winding. It never goes where you expect, and it's okay as long as you're happy with the end result. So thanks very much for listening.
Thank you very much, Sarah. Hello. Thank you very much, Sarah, for such a lovely talk, your own personal story, as well as the life cycle of something which is scientific, a product which is scientific. Lots of interconnected bits of physics, chemistry, biology, mathematics, computer science, all of these things all feed in to form something which is quite innovative and new and could have real patient outcomes. We have a QR code for the Slido Q&A system.
I'll take off the picture of my lab running away from me. They do it all the time.
So there have been a few questions already, so I'll get started with those. But if you want to scan, if you have any questions, please do scan, type in the Slido number, ask your question if you so wish. We'll go for a few questions, and then with mind on the time, those of you, if we can't get round to your question, we can take questions afterwards, okay? So I'll take some initial questions first. Question number 1 was, are the patients unconscious for this whole process?
Yeah, typically in endoscopy, you're sedated, yeah.
Okay. Is it painful?
I have never had one myself. I have been told, my husband has had them, I'm told they're uncomfortable because you're sedated. You're not unconscious, you're sedated. So you are conscious for the experience, but you are given sedatives. So as I understand, not painful, just uncomfortable.
Okay.
Okay. We have a living proof. Yeah, it's very fun to watch people doing medical procedures. I enjoyed watching the ultrasounds when I was pregnant.
Okay. How early could these techniques pick up potential issues? Would there be any symptoms at the stage that would prompt a doctor to check a patient?
Yeah, that's a really interesting question. In patients who are diagnosed with Barrett's esophagus, they actually routinely go for endoscopy every 3 to 5 years. The critical bit is being diagnosed with Barrett's esophagus. So many people in the country actually have this disease but are not aware that they have it. It's commonly confused with chronic reflux. People are just given antacids and sent home in order to treat reflux, whereas in fact it's masking an underlying condition of Barrett's. A colleague of mine in Cambridge, Rebecca Fitzgerald, has recently invented a really exciting new device, which is effectively a tiny scourer which you wrap up in plastic and put on a little string. And people can swallow this in a GP's practice, and as it's pulled out of the esophagus, it captures millions of cells from the esophagus, which can then be analysed and allows them to detect Barrett's and discriminate that from standard reflux symptoms. And there's now a 4th national trial looking at the efficacy of this as a way to detect the risk factor of Barrett's, and that would be a pipeline for people to then be coming in for endoscopy to look for those early signs.
Do you have an estimate for how long it will take for this all to become mainstream?
In my ideal world, in my lifetime, I would like to think that the data that we acquire now would be potentially impactful on a sort of 5 to 10 year time frame. But that largely depends on the rate at which you commercialise the device into a product that can be sold and adopted into healthcare systems. So what I'm doing in the lab is asking one of my students to go and build something ad hoc, and then maybe go and build another thing and build another thing. That's not a reproducible manufacturing process. So you need to get out into a commercial environment that's appropriately medically certified and qualified and regulated in order to then be used in a setting. But there are endoscopy companies out there that are looking at spectral information and its potential, and we're working with some of those with the view to get this sort of idea into clinical practice.
Beyond the digestive tract, could this be used in other scenarios?
The endoscopic manifestation could also be used, for example, in the lung. It's also been thought about in other kind of minimally invasive approaches like laparoscopic surgery, where you do surgery in an endoscopic setting. You may have heard of the big surgical robots that are now being used, where they're doing remote operations with the doctors controlling them almost like a video game in another room, and the patients being operated on by the robots. There are active research activities looking at using spectral colour information in those settings as well.
Okay. Should doctors know how the light filter system works in order to help them find polyps better?
Well, one would argue yes. Most doctors are not so concerned with how the thing works, but that it does work. And the ability for it to give them useful, interpretable information is going to be the key criteria in getting this adopted. For it to be easy to use, easy to integrate into the current workflows, and economically viable relative to the existing standard of care.
Okay, thank you. And a few questions on education. What are good courses for uni if you're interested in biophysics? And allied with this, what was your deciding reason to choose natural sciences at Cambridge?
Okay, so in terms of decision making, I came from a physics background and learnt the biology when I got there, and I would advocate that approach rather than going down the biological route and learning the physics later. I think physics is much better done with routine practice, as we've heard. Lots of Isaac physics problems, lots of routine practice. Learning to become a physicist and then applying that, I think, is a really good route. So I would advocate going for a very strong, broad physics education as an undergraduate, because you never really know where you're going to end up. And a lot of, for example, biophysical research is looking at, for example, fluid dynamics in the cilia of the lungs. And so there you need a whole range of different physics principles that you wouldn't get if you specialised immediately. In terms of applying to natural sciences at Cambridge, I was pretty confident that I wanted to be a physicist, and that was actually more a practical decision, which was simply that in the sixth form college I went to, it wasn't a big feeder school for Oxbridge, and there was the odd person here and there over the last decade who'd been, and someone had been to Cambridge, so I thought, yeah, I'll go there, because someone else has been there.
What kind of activities, projects would you recommend people do if they're thinking of doing something similar to your research?
I think regardless of whether you're thinking of doing something similar to my research or some completely different research topic, the number one thing I would recommend you do is become proficient in Python. Learn how to code well, and code regularly, because anything you do will involve coding. If it's computational simulations, all the simulations we run are in Python, all of the data analysis we do is in Python, all the data acquisition we do is with Python-controlled devices. I chose Python in my lab because of many open source reasons. We do a lot in terms of reproducibility of research, standardization of research, and open source, open access work. But in general, if you can code well, code efficiently, it will help you enormously throughout your undergraduate degree in terms of your experimental classes, and it will serve you well for your career to come. So spend your summer learning Python if you don't already know it.
And there is the BPhO Computational Challenge, so please enter. Do you think remaining in education for as long as viable was beneficial to your career?
I mean, as an academic, I would have to say yes, because doing a PhD is almost a prerequisite for coming into an academic career at the stage when I was entering. I would have to say these days the boundaries between academic research and industrial research are much more porous. So there are many people working in an industrial research setting that come into academia and people who go out from academia. However, regardless of the setting that you're doing your scientific studies in, whether it's a research environment in a company or whether it's a research environment in a university, the sorts of skills that you will learn by doing postgraduate research in a PhD are hugely translatable. Even the broader skills, like being able to write effectively a book about your work, shows incredible diligence in report writing, the ability to construct scientific presentations, and go out and communicate your research to others. All of these broader skills, in addition to the scientific and data-driven skills that you'll develop, are great. And so most of these PhD research topics will come with stipends, which will provide you with some living expenses. And things that I did, for example, during my PhD, I went out to work in schools and I went to do tutoring in order to earn additional money to make it a sustaining career choice rather than, let's say, an economically unviable choice.
Okay, a final question before we end for today. How do you think future advancements in AI will affect early diagnosis of fatal diseases like cancer? So very wide question.
Yeah, it's very wide. I will try not to spend 20 minutes answering it because you'll probably drag me off the stage. We've already seen huge efforts globally in using AI for early cancer detection. A good example is in mammography, where we have huge databases of women who are being screened regularly for breast cancer, and each of those screens is normally individually observed by a human. But the vast majority of those screens are negative. So there's been a huge amount of work recently looking at training algorithms to confidently give a negative result. When you get to a positive result, there's a whole interesting swathe of social, anthropological, ethical questions that come about whether you want a computer to diagnose you or not. And some people are very much in favor of having a computer diagnose them, and some people are very much not. But if you could have a combination of a computer that flags, these are the slightly unusual scans. So now the radiologist who has to look at the scans, instead of having to look at thousands of scans over the course of a year, now only has to look at a few hundred. They'll have more time to spend on each of them. They'll be better at interpreting them. There's also ways that the computer, like the AI interpretation, can inform the clinician. sort of thing that I was describing in terms of giving colour mapping to a clinician. Ultimately, if the person is doing a procedure, let's say a surgical procedure, a colour mapping of an AI interpretation might be sort of look here, or look there, or resect here, or ooh, you missed a bit. Those kinds of computer-aided tools are becoming, very much widespread. There are several of them that have been approved by regulatory bodies and are already used in patients, and I think this will only get more and more prominent. The key, of course, with all of these is having good, well-curated, well-annotated data sets to train them, because the old adage of garbage in, garbage out still holds. So we have to be very cautious about applying them and interpreting them, but I think it will be a huge growth area for the medical industry.
Yeah, and areas that you students, those of you in the audience, are very much at the forefront. So I hope today was an interesting day. So well done again for the prize winners. And those of you who've joined just for the lecture, I hope this was an interesting, starting point for where you might develop your own scientific interests. Thank you again, everyone, for attending. If you have any further questions, do come up, okay? We'll be available for question and answers for a few more minutes. Those of you who want further refreshment, there is tea and coffee in the room next door where we had lunch. Thank you again.